So I'd now like to welcome our next speaker. So our next speaker is Nachiket Kashikar, and Nachiket will speak to us about every cell matters, molecular cartography, and using that to study changes in gene expression in Alzheimer's disease at the single cell level. And Nachiket is speaking to us from Resolve Biosciences, which is based in Germany, I believe. So when you're ready, we can see your slides. And so I think you need to unmute, but then when you're ready, please go ahead.
Yep, many thanks, Joe. Good afternoon, everyone. Can everyone see the slides and hear me?
Yeah, looks good to me.
Excellent, cool, thanks. So first of all, thank you for the invitation, and it's great to be part of this meeting, which is really dedicated on spatial methods, and particularly in the context of neurodegenerative diseases, or let's say neuroscience. And as Joe rightly guessed, so we are based in Germany, let's say about 10 kilometers from Düsseldorf. So in the next to the Rhine, actually. And we are a spin out of QIAGEN. So the technology was developed at QIAGEN a few years ago, and we spun out into a completely distinct entity early last year. And so essentially we are at the moment commercializing our, let's say, spatial transcriptomics or molecular cartography platform. And as you can imagine from the title, so what I've written here is like every cell matters. So as you can probably rightly guess, so we are looking really at single cell, and even at subcellular resolution. And so there are many different approaches when it comes to doing spatial transcriptomics. So like you have sequencing based approaches, and you have approaches where you're doing essentially FISH and so on. So our approach is essentially based on FISH, single molecule FISH. And just to give briefly an idea about cartography. So if you look at traditionally, so the cartography is art and science of geographically representing and charting a geographical area. So we aim to that in the context of tissues, biological tissues, and any kind of tissue in principle. So as you can see on this slide, so what I'm showing is actually zebrafish, which is zebrafish retina on the left side, zebrafish brain on the right side, and in the middle you see actually dragon brain. So that's the bearded dragon, Pogona viticeps. So what I'm alluding to is that the technology that we have developed is not limited to a given species. Of course, when you talk about neuroscience, neurodegenerative disorders, and whether you're talking from academic or pharma perspective, you're always going to work with mouse as a model system and also human clinical samples. So most of my presentation will be on the mouse data set and also a little on the human data set, but the technology is not limited as such when it comes to sample types. The other thing that I would like to really highlight is that when it comes to analyzing the spatial transcriptomics, or let's say the molecular profile, so one key aspect is sensitivity. And I'm going to spend some time to talk about the sensitivity aspect of our technology and also compare it to some of the other technologies. So my talk is going to be mixed of, let's say, both technology and a bit of neuroscience. So let's start with mouse brain. So I mean, that's one of the favorite model systems when it comes to spatial, right? So what you're seeing essentially is the mouse coronal section. On the right side is the impression from the Allen Brain Institute map, really depicting different brain regions here. And what you're seeing on the left side, where we essentially perform molecular cartography using 100 genes. And so we have covered, let's say, cortex, hippocampus, and some other parts of the brain here. Now we are going to dive deep into this data set. So what I'm going to do is essentially show you the power of the technology, do a case study in the context of Alzheimer's mouse model, and then take to the human data. What you're seeing on this slide is marking of cortex based on a few different genetic markers. So we are focusing, let's say, for example, on carbindin 1. So that's expressed, so that's represented in the red. So as you can see that you can nicely see expression of carbindin in layer 2, 3, and also sparsely in layer 5, 6. So that's something that you would normally expect, right? And cortex, that, as you know, mouse cortex, it has 6 distinct layers, and we beautifully capture these 6 distinct layers purely based on the genetic markers. Now we compared our data just to be 100% sure about our specificity and sensitivity to some data that was published by Chris Ponting's group back in 2011. So it was done using ISH. So what you are seeing here is a few markers, of course, CUX1, CUX2, and so on. That was done using ISH, and this is our data for the same genes. Of course, we did that using a panel of 100 genes. What you can nicely see is that wherever CUX1 is expressed in ISH, we exactly see expression of CUX1 in the same layer. Same is for CUX2, and same is for all the other genes. And just as a case in point, if you look at, let's say, CTGF, so that's expressed in a very thin layer, 6B, and we pick up exactly 6B for the expression of CTGF. So what I'm saying here is that when it comes to specificity, the technology is highly specific. Now, to have a measure of sensitivity, we compared our data with bulk RNA-seq. And again, as you can really nicely see, the correlation is 0.9, so it's quite strong. Now we are going to look at different cell types. So as you know, brain is a very complex system, complex tissue, and really fascinating to study, whether it is in development or health or disease. And what happens in, let's say, health and disease is change in different cell types, right? And brain has different cell types, neurons, astrocytes, and as Omar was nicely pointing out, different subtypes of astrocytes and so on. I'm not going to the subtype specific story here in this presentation, but what I would like to show you are, let's say, different glutamatergic neurons here, which are presented in red, GABAergic neurons, microglia, which are green, astrocytes, which are magenta, interspersed here in the tissue, and oligodendrocytes. Now we are going to zoom in on, let's say, inhibitory neurons. So one of the very important cell types in the brain, right, to control the excitation and inhibition balance. So we are looking at GAD1 positive cells, parvalbumin positive cells, somatostatin, and VIP, right? So for example, if you just look at the yellow cells, so these are somatostatin positive neurons. And again, if you look, for example, VIP, so these are really rare cell types, and we also pick those guys very beautifully distributed in the cortex. Now, let's say, for example, so typically we see any spatial data in, let's say, nucleus and something around it, right? So what you can also beautifully see is kind of expression in the processes, right? So for example, where my mouse is, the cursor is moving, you can also see the expression of this specific somatostatin transcripts in these processes. And now this is kind of the 3D representation of that particular cell. So yellow blobs are somatostatin RNA molecules. So there are about 322 in total, and all the others are different transcripts in this case. So why you see that 3D representation is that we start with a 10 micron thick tissue section, and we take multiple Z planes throughout that depth of that section, and we have these spot coordinates for in X, Y, and Z. And typically, of course, we present it in 2 dimensions, but if you want to make use of that data in 3-dimensional space, that's completely feasible. And that's kind of the power of the technology. And again, just to focus on these animations, of course, the blue dot that you see in the center is nucleus, as they were using DAPI, and you beautifully see all the spots surrounding the nucleus here. Now, what we have done here is use DAPI as a proxy for cell segmentation. Of course, when it comes to cell segmentation, it's a vast topic, and we are not going to touch upon that here, but of course we can discuss that in the panel discussion. But what we have done here is we took each DAPI as a proxy, and we drew boundaries 8 microns around the periphery of each DAPI signal. And we use QPath to do so. And we have segmented here about several thousand cells. And then essentially what you get is cell by gene matrix and gene to cell matrix. And then, for example, you can start to do some basic statistics, like how many signals are you getting per cell, or how many genes you are going to detect per cell, and so on. And then we did clustering of about 35,000 different cells for all those 100 genes. And we beautifully pick up distinct clusters for neurons, astrocytes, and so on. And those clusters are projected back onto the tissue. And that's the animation that you see on the right side, right? And you can now start to look at these different clusters in detail. For example, all these spatially segregated cell subtypes. So for example, what you see in the top right are glutamatergic neurons, then astrocytes, oligodendrocytes, microglia, and so on. And these are essentially the clusters projected back onto the tissue. And then we, of course, counted the fraction of cells that you see. For example, we have 53% glutamatergic neurons, about 10% in GABAergic neurons, and so on. But of course, you get that information in its full spatial glory. I'm just trying to move to the next slide. Right. So this is just to this was just to show you the power of the technology when it comes to analyzing the brain tissue and specifically the mouse tissue. Now we took exactly the same approach and implemented it in the context of Alzheimer's disease. So this is in collaboration with Janssen Pharmaceutica in Belgium. And what we did is we took APPPS1 as a mouse model. It's an amyloid mouse model, of course, and it is of course going to be full of plaques at a certain age. And the central question we are asking is how does the gene expression change as a function of distance from the pathology? And in this specific case, so we had, of course, cell type specific markers and also genes for 4 different signaling pathways. One of them was, of course, disease associated microglia, Wnt signaling, and so on. Now, if you just let's say take a bird's eye view, right? So what you're looking in the left half of the slide is cell type marking. So on top left, you have wild type mouse cortex, and different cell types are labeled in different colors. For example, neurons in turquoise, oligodendrocytes in red, and so on. If you look at the bottom left panel, you see the similar cortical area, but in the presence of plaques, right? And you can just immediately see that there is a dramatic or drastic reduction in neurons, right? So you see you see fewer of these turquoise blobs essentially. What you also see here in the APPPS1 cortex are these yellow blobs. So these are essentially amyloid plaques. And in this case, we labeled the plaques using a dye called PFDA. And this labeling was done after performing molecular cartography. So again, the point of the matter is that the tissue remains fully intact after performing molecular cartography so that you can use additional dye labels such as DAPI, PFDA, or you can also in principle perform antibody staining. Now, what you see on the right side, it's the same tissue, it's the same section, but what we are showing essentially are different genes. So red guys are essentially enriched in the wild type cortex, and the green guys are essentially enriched in the APPPS1 cortex. Now we are going to look at it in a bit more quantitative detail. What you see on the left side is RNA-scope data actually. So this comes from Janssen. It highlights 2 different genes, CST7 and TREM2. And what you nicely see here is that there is enhanced expression of CST7 and TREM2 in the close vicinity of the plaques. Of course, one of the problems of using RNA-scope is that you cannot really do any sort of quantification because it's an amplification-based technology and the blobs really appear like it's like a smudgy thing, right? And of course, the other issue is that you cannot do multiplexing. We essentially observe exactly the same thing, reproduce that data, but now you can start to really quantify how many molecules of TREM2 are in the close vicinity of the plaque and how they how does that, let's say the expression changes as you go away from the plaque, right? So what we did is we took each plaque as a center. So there are about 635 plaques from 3 different animals, 3 different tissue sections. We took each plaque as a center and then we drew concentric rings of equal area around these plaques. And then we essentially counted transcript numbers in rings with equal area around the plaque, as I said. I'm just showing here example of 4 different genes, C1QA, CST7, SIT1, and TUBB3. So if you, for example, just focus on C1QA and CST7. So CST7, I already show you here the raw data. So CST7 is really highly expressed in the vicinity of the plaques, and as you go further away from the plaques, expression goes down, and the same is for C1QA. On the other hand, the expression of SIT1 is quite low when in the vicinity of the plaques when compared to away from the plaques, right? And we have done that for all the 100 genes in the panel. And at the moment, I cannot show you all the data because we are finishing up a manuscript essentially, but just to give a sense of that, right? So you can start to also do clustering. What you see on the left side, on the left panel, is essentially UMAP clustering, which is a mixed clustering of wild type and APPPS1. And again, you beautifully pick up different cell types. In the middle panel, we have sort of segregated that based on the pathology, right? And on the right side, on the rightmost panel, so we are segregating it based on the distance closest to the plaque. And blue is the closest to the plaque and red is further away from the plaque and so on. So again, if you go back to the middle panel, you can nicely see 2 distinct clusters in microglia, same for astrocytes and so on. Now, of course, we did further quantification in this specific case. So we quantified the number of glutamatergic neurons, both in wild type and APPPS1 cortex. We see dramatic reduction in the glutamatergic neurons, and on the other hand, we see substantial enrichment in microglia. Now, where does that occur, right? So that's the most important thing, where? So we find that in about, let's say, 20 to 30 microns in the vicinity of the plaques, all the changes happen. For example, you have really enhanced expression of microglia about 20 microns, let's say within the sphere of 20 microns from the plaques. So what I'm saying here is that the sphere of influence, so to say, for each plaque is about 20 to 40 microns. Now we can also take the next steps, right? So what we are seeing here is shifts in the population of microglia and astrocytes. So on the left side, you have APPPS1, and on the right side, you have wild type section. What we are showing essentially are two different cell types, microglia and astrocytes. And specifically, when you look at microglia, there are homeostatic microglia and disease-associated microglia. So if you look at the wild type tissue section, so you see a lot of homeostatic microglia nicely dispersed throughout the tissue. If you look at homeostatic microglia in the epiphyseal cortex, you hardly see those, right? You of course have some, but you hardly see those. But most of them are converted into disease-associated microglia, and those can be seen in really this bright green color. And mostly they are enriched in the close vicinity of the plaques. The same is essential for astrocytes. So you have you don't have many reactive astrocytes in the wild type cortex, but you have reactive astrocytes dispersed throughout the tissue, which are essentially labeled in dark red when it comes to APPPS1 cortex or APPPS1 tissue. So that was about, let's say, the mouse tissue. And I would like to briefly show you some data on the human tissue. This is still early data, again in collaboration with Janssen. The tissue was obtained from the Dutch Brain Bank. So we have, of course, post-mortem brain tissue sections obtained from BRCA6 patients in this case and age-matched controls. What you're seeing here essentially is comparison between the two. Okay. Different cell types are labeled in different colors. So for example, this turquoise is also labeling neuron and neurons based on, let's say, markers such as SNAP25, SLC17A7, and so on. And again, there are different cell types like oligodendrocytes, astrocytes, and microglia. Again, if you just take a bird's eye view, you can see that there are hardly any neurons left in the BRCA6 tissue, which is kind of a really stunning finding. Of course, it's not a new finding, but when you start to look into the spatial context, it's really amazing, and it tells you what's really happening in the Alzheimer's tissue. Of course, we are now taking this study forward exactly the same way as we did the mouse, because again, in the BRCA6 tissues, you are going to have both pathologies, amyloid and tau pathologies. So how both pathologies impact the expression of different genes and also different cell types. So that's something we are investigating at this stage. And just to kind of again give you an example, so here we have clustered the segmented cells. Again, the segmentation was based on DAPI, clustering was done using CRAT, and again we find beautifully different clusters of oligodendrocytes, neurons, and so on. And again, we are distinguishing those clusters between healthy and BRCA6 stages. Again, for example, if you specifically look at microglia, you see two really distinct populations. For example, the healthy population is in green, and in reddish, it's the BRCA6 population. So just to give you a brief final argument about final data set on the human tissue. So we compared also the data set with the in situ sequencing data that was published recently. So we obtained the data from the website, which is highlighted here. So we compared actually molecular cartography to the ISS-based technology. These are non-disease human brain samples, so there is no pathology in this case. We have selected only neuron-rich regions of equal size, and the segmentation is of course DAPI-based. We had about 28 genes common in both data sets. For example, for neurons, we had CYT1, oligodendrocytes, MOG, and so on. So what we are looking at here is roughly equal area, roughly equal amount of cells, about 2,000 cells in both cases. On the left side, what is actually the ISS data or in situ sequencing data, and on the right side is the results molecular cartography data. What we did essentially to compare the sensitivity is we counted the number of transcripts per cell. And what we find is actually stunning. So the mean transcripts per cell with the ISS is about 0.15. And the number of mean transcripts per cell with using molecular cartography is 100 times. It's actually quite several orders of magnitude more. It's about 138. So it's quite stunning because that also highlights in terms of choosing the panels for your experiments, right? So if you want to define a cell type, you do not need 10 or 20 genes if you are sensitive enough. So if you are sensitive enough, you can reliably assign the identity of that cell just based on 2 or 3 different markers. And just a bit of last data set on sensitivity. So this is done in cell cultures. This is an in-house data. And just a glimpse here. So what we have done is compared our technology with conventional single molecule fish done using just one gene at a time. And I'm just showing 2 extreme cases when it comes to expression. So APC, so that's one gene, and CNPF. So APC has relatively low counts per cell, so about, let's say, 10 or so. And for CNPF, it's about 10 times more. In both cases, the, let's say, counts per cell that we find using our technology is almost identical to conventional single molecule fish. So in other words, if in using conventional single molecule fish, you are going to identify 100 copies of a given mRNA in a given cell, our technology is going to identify and detect and quantify exactly the same number of molecules for that gene in a given cell. So I think at some point of time, all the different technologies need to come to a point where we have those quantitative measures for sensitivity and other parameters. So just to wrap up in the last, let's say, 3 to 4 minutes or 5 minutes, so I would like to briefly highlight what Resolve is offering. So what you're saying is that our differentiation is purely based on methodology, and I will tell you briefly about the methodology. So highest sensitivity to detect gene regulation, and I showed you some data when it comes to sensitivity. The second thing is cellular resolution for all cell-specific transcript analysis. And I also showed you the power of technology when it comes to really looking at subcellular locations, right? So I did not really make a scientific case out of it, but the resolution is such that you can really identify transcripts in different subcellular compartments. And the third part is tissue integrity for analysis of environment-specific gene transcript changes. So for example, I just showed you, you can label the tissue with using additional dyes, or you can do antibody staining and so on. And that's something we think at Resolve that really differentiates us from some of the other technologies which are based on laser captured, in situ sequencing, or amplification based fish. Briefly, how does the technology work? So it is based on combinatorial color coding. So you typically start with the cross section. The section is 10 micron thick, and it is stuck onto the glass surface. The panel of and then you of course design the gene specific probes. There are multiple probes that bind to the gene. We color the probes using fluorescent molecules, and then we image the samples and we decolorize the probe. Now, if you are doing 100 genes in an experiment, it does not mean that we are repeating this cycle 100 times. So it happens only a few times because we had this combinatorial aspect here. Of course, combinatorial aspect is implemented by some of the other technologies also, but how that combinatorial process works, so that's essentially Resolve's proprietary technology. And I'm happy to talk about that, about exactly the sample preparation, etc., in the panel discussion. So just to give you the summary or the gist of what molecular cartography is offering you. So we do up to 100 genes simultaneously. There is in principle no limitation which genes you can or cannot choose. The panel is completely customizable. Of course, we have certain panels in-house when it comes to brain tissues, whether human or mouse, or also even different other tissue types. So there is in principle no limitation. Of course, we tend to avoid genes which are really highly expressed. As you can imagine, the technology is based on imaging. So each spot is diffraction limited. So we tend to avoid genes which are super highly expressed, but in principle, let's say 98% of the genes do not do not offer this problem. The second part is resolution. So cellular and subcellular, I showed you the data. And again, I would like to reiterate the 2-dimensional and 3-dimensional aspect. So we have information throughout that 10 micron thick tissue. And if you want to have that 3D rendering of the tissue, that's perfectly feasible and possible. Of course, typically you present information in 2 dimensions. Of course, you are generating a lot of data. It's an imaging based technology. And again, as a user, you do not want to start analyzing all the raw image files and so on. So we have dedicated bioinformatics pipeline, which is an interactive software. So what you get essentially is let's say the XYZ coordinates of all the million spots that we detect, the identity of each of the spots, the DAPI signal, then cell by gene matrix and gene by cell matrix, then where essentially the user can start to analyze the data in great detail. In the last bit is digital quantification. So each spot that you see is one transcript, so you can really quantify. Multiplexing, we talked about it. We can combine it with post-hoc cell labeling using dyes or immunohistochemistry. Important is throughput. So quite often you are limited to 1 or 2 tissue samples on a given slide, but we can run up to 24 samples in parallel. So those 24 samples are distributed over 3 different slides, and we can again talk about it in detail if you're interested. Sensitivity, we talked about it, and I showed you the data. And the reason behind really exquisitely high sensitivity is we do not use any enzymes. There is no amplification related bias, there is no capture required, and we really get rid of all that stochastic and loss of, let's say, the low efficienciness of all those methods. So we do not have that. When it comes to sample types, again, from the neuroscience point of view or the neurodegeneration point of view, so human tissues, mouse tissues, that's perfectly fine. There are no issues there, specifically when it comes to brain samples. Cell culture, that's really easy to go. There are absolutely no issues. Neural cell cultures, iPSC-derived cultured neurons, even organoids. So we already have a white paper published on our website. If you think that there are other organs involved in neurodegenerative diseases, maybe you find links to liver, spleen, we are all good there. So what I'm saying here is that it's a really versatile technique. And if you're really interested in trying the technology out, so we are launching the commercial service now. Please reach out to the contact highlighted here. Thank you very much. And I'm open to take the questions.
Great, thank you very much. Thanks, Nachiket. So there's a couple of questions that have come in. So the first one is from Blanca Diaz Castro, who says, "Hi Nachiket, can this technology be combined with immunohistochemistry?" I think you've touched on this.
Yeah, absolutely. So again, you have to be careful. Of course, technically and theoretically, of course, you can combine it, but it depends from antibody to antibody.
Sure.
So antibody needs to be validated and so on. Again, sample preparation methods can be different, whether it is fresh frozen, packaging fixed, or FFP, and so on. So in principle, yes, but of course, we go case by case here.
Yep. Okay, thank you. There's also a question and a comment from Malta, who spoke earlier. So he says that you are comparing your technology to the old version of ISS that is cDNA based. So that's his comment. I'll move on to his question.
Sure.
What do you believe is the reason you see more transcripts than single molecule fish?
So we do not, right.
Go on.
So, sure. So we do not really see more transcripts compared to single molecule fish. Of course, I showed you representative data. And of course, you have to give 5% here and there. So we are doing a lot of statistics on this using different cell culture systems and also in tissues. So based on the data that we have available at the moment, so we are, let's say, in the range of 95 to 100% when it comes to single molecule fish.
Okay, thank you. I have a question, and I really like towards the beginning of your talk how you can see expression in the neurites. I guess my question is, it looked from the image you showed that it was only the neurites that are still relatively close to the cell body. And bearing in mind, let's say in the mouse cortex, you can have an axon that's 1 millimeter long. How far out can you go?
So, I mean, we don't have that resolution to really track the neurites. That's absolutely sure. Of course, we are not doing any expansion and all that stuff. So actually, I was surprised that even we see this sort of stuff. So it was really pleasant surprise for me. I mean, my background is neuroscience. I was really happy with this. And maybe the density of the spots is quite high, closer to the cell body. That's one of the possible reasons. Yeah. So if you really want to trace axons and dendrites, I think you need to have expansion to a certain degree, but we are not claiming that at this stage.
Yeah. Okay. Thank you. Great. Okay. And the other, quick question was that when you showed that most of the changes occur within 20 microns of the pack, which fits in with what others have shown about synapse loss around plaques, that kind of thing. My question was, you measured that from the center of the plaque, right? Not from the edge.
Right. Yeah. So absolutely. So I mean, the thing is, of course, plaques come in different sizes, right?
Exactly.
And again, the data that I showed you was from APPPS1, which was of certain age, and then let's say 7 to 8 months and so on. So there are going to be variations when you look into the patient data where, again, you have microplaques. How does that influence? So that's something to be discovered together with the UK DRI people.
Yeah. Okay. And then lastly, so then if someone wanted to get in touch with you, I guess they could just email you straight up and work out if they wanted to work with you.
Yes, absolutely, absolutely. Just drop me an email. So it's my name, Nachiket.Kashikar, and at resolve.biosciences.com. I can also put it in the chat. Yeah. And or just visit our website. Looking forward.
Lovely. Thank you very much. That was great. Thanks.